Welcome to Unical Pre-Degree Digital Lecture Series. My name is Dr. Emmanuel Efan, and today I'm going to be taking you on the supporting tissues and movements in living organisms, both plants and animals. So plants and animals, and indeed all living organisms, need tissues and other supporting systems to enable them maintain their shape and structure. In plants particularly, these supporting tissues give them definite shape, strength, rigidity, and resistance against forces like wind and water. So the main supporting tissues in plants include parenchyma, collenchyma, sclerenchyma, and then the xylem and phloem tissues, which are also the vascular bundles. Here's a diagram of the three major plant tissues, supporting tissues. We have the parenchyma, both the lengthwise shape of it and the transverse section of it. We have collenchyma and then sclerenchyma tissues. So we look at the properties of each of these supporting tissues in plants. The most common and abundant plant tissue is the parenchyma tissues. So they are found everywhere in the plant, from the cortex of the stem to the phloem, in the roots, leaf, mesophyll, etc. And they are made up of living cells. Those cells are thin and have large vacuoles with many air spaces between them, as you can see from the diagram there. You can see the intercellular spaces. Now, parenchyma tissues are responsible for the firmness and turgidity that we notice in plant stems. In addition to that, they also synthesize food in the leaf and can also store water. The next supporting tissue, collenchyma tissues, are usually located in the cortex of the stem, roots, and in the hypodermis. They are composed of living cells also, which are elongated, flexible, and thickened at the corners. From the diagram, you can see those characteristics. They are responsible for strength, support, flexibility, and resilience that we observe in plants. The third supporting tissue, the sclerenchyma tissues, are found in the pericycle of the vascular tissues and cortices of roots and stems. They are composed of cells with thick walls containing lignin in addition to cellulose. There are two types of sclerenchyma tissues, and these are fibers, which are elongated cells with pointed ends, and then sclerids, which are not elongated but provide strength. Now, the xylem tissue is also a supporting tissue, though we will still discuss it under vascular bundles. They are found mainly in the vascular tissues of the root, stems, and leaves. They provide support, strength, and shape to the plant. But particularly of importance is that the xylem tissues conduct water and mineral salts from the roots through the stem to the leaves for photosynthesis. And then the xylem tissues are in themselves made up of tracheids, vessels, fibers, and xylem parenchyma. Phloem tissues are similar to xylem tissues in that they are also part of the vascular bundles of plants found from the roots to the leaves. They are made up of sieve tubes, phloem parenchyma, companion cells, and phloem fibers. They provide support to the entire plant, and in addition conduct manufactured food from the area of synthesis, which is the leaves or the green portion of plants, to areas where they are needed or stored. Now the mechanism of support in plants is as follows. We'll start from the outermost layer in a cross section of a typical stem, a typical stem. So on the outermost layer we find the epidermis or piliferous layer. The epidermis is the outer covering of the leaves and stem, while the piliferous layer is the outer covering of the root. So epidermis is the outermost covering of the stem, piliferous layer is the outermost covering of the root. Now this epidermal layer is only one cell thick and functions in protection and prevention of loss of water, amongst other things. So from the diagram there, you can see epidermal hair, epidermis, so that shows you that it is a stem or any other part of the body, but not the root. Now beneath the epidermis, we find the region called the cortex. So it is between the epidermis and the vascular bundles, particularly in dicotyledonous stems. The cortex is made up of collenchyma on the outermost part, and then parenchyma in the middle region, and the innermost area is the endodermis. Now the collenchyma and the parenchyma in the cortex provide strength and support, while the endodermis serves as starch stores starch products. Next to that, in a typical plant, we will see sclerenchyma, which is a layer on the outer part of the vascular bundles. Now sclerenchyma is made up of dead or lignified cells that contain lignin. It acts to strengthen the stem or give mechanical support, in addition to giving flexibility to the plant also. Now after these regions, we now move to the vascular bundles, which are the inner part of the plant. So they are found in the inner part of the stem and consist of xylem, phloem, and cambium. Like we mentioned before, xylem conducts water and dissolved mineral salts from the soil through the roots, through the stems, branches, and so on to the leaves. Phloem, on the other hand, conducts manufactured food from the leaves, which is the area of synthesis, to every other part of the plant where the manufactured food is needed or for storage. Now, cambium is found between the phloem and the xylem. They are constantly dividing cells, and when they do so, they also increase in size, providing secondary thickening. This is the main reason why we see tree trunks increase in their thickness over time. Now, the innermost part of the plant is the pith. It's the central part of the stem. It is made of parenchyma and extends between the vascular tissues. So the diagram there shows you all these things that we've talked about. Now, in flowering plants, strength and rigidity are achieved by a combination of these supporting tissues and also turgor pressure. So, for example, the parenchyma cells of the pith, when they are fully turgid, that's when they absorb water and they become round or full. In other words, turgid. They push outside with a force, and then this force is also restrained by the inelastic epidermis, so making the plant firm. So in other ways, we are saying that parenchyma tissues, because of their turgidity, they provide hydrostatic support to the plant. Here is a comparison of the transverse section of a dicotyledonous stem and a monocot stem. So dicot stem is on the left hand side, the A, and then the monocotyledonous stem is on the right. We can see how the vascular bundles are arranged in the dicot stem and how they are scattered in the monocot stem. The lower picture there just shows an enlarged sector of each of the stems. So there you can find all the structures we've talked about: epidermis, cortex, vascular bundle, pith, cambium, endodermis, parenchyma, etc. Now we take a look at the transverse section of a dicot root. We can also see that the piliferous layer is the outermost layer. Then we see the cortex, the phloem, and so on. Then when enlarged, you can see the structures more clearly. Also, we see here the transverse section of the monocot root with an enlarged section showing the different features and supporting tissues. Here is the transverse section of a dicot leaf also, showing its supporting tissues. Primarily, you can see the epidermis there. You can see the vascular bundles grouped together, and then you see the guard cell surrounding the stomata, which opens and closes during transpiration, and so on, and other features there. So we did talk about secondary thickness being given to plants. This is a clear example. The first diagram there, the first one shows the structure of the supporting tissues, and then when you come to number 4, you can see that over the years there's a secondary xylem that has developed. A secondary phloem also has developed with a cambium ring responsible for that, and then year after year the tree becomes wider in width. So with the supporting tissues understood, the other responsibility of that is that it aids in movement in living organisms. And though we've not talked about animals yet, we'll just finish up with plants, and then we'll now do the same thing for animals. So when we're talking about movement, generally we observe that animals respond more quickly, or they respond quicker to external stimuli than plants. And then when animals do, the entire organism responds. But in plants, it is slow, and then only certain organs of the plants respond. So generally in biology, there are 3 basic kinds of movement or responses. And these are tactic movement, nastic movement, and tropic movement. We're going to look at each of them in a bit. Taxis or tactic movement. It's a directional type of response. In this case, the whole organism moves in one direction in response to the stimuli. So the movement could be positive or negative. So you can have a positive taxis or negative taxis. Positive if the organism is moving towards the direction of the stimuli, and then negative if it is moving in reverse away from the source of the stimulus. So examples of tactic movement are thermotaxis, that's in response to temperature or heat; phototaxis, in response to light stimulus; hydrotaxis, response to water; and chemotaxis, in response to chemicals. Here's a simple diagram showing tactic movement in Chlamydomonas. You can see there, it moves towards light, and then it moves towards warm water, and then during reproduction you can see another organism there, how the male gamete moves to locate the female gamete so that sexual reproduction can take place. All these are examples of tactic movements. Next, we consider nastic movement or nastism. This is a type of response in which only part of a plant moves in response to, this time around, non-directional stimuli, such as changes in light intensity, temperature, or humidity. So example is a mimosa plant, a plant that folds when you touch it. And if you've ever experienced it, you see that it's not the entire leaf of the plant that will fold. It's only the part that has been touched. Now the next kind of movement is tropism or tropic movement. This is a type of response in which a part of the plant moves in response to a directional stimulus. So it can also be positive or negative depending on whether the plant moves towards the source of the stimulus or away from the source of the stimulus. So tropisms are very slow growth movements. Examples are phototropism, that's if it moves towards light, hydrotropism towards water, geotropism in response to gravity, chemotropism, chemical substances, and then thigmotropism in response to touch, like the mimosa plant. So here is a picture showing those different tropic movements in plants. You can see phototropism, thigmotropism, hydrotropism, and so on and so forth. So we've succeeded in explaining and describing the supporting tissues in plants and the different ways it helps the plants, particularly in giving them rigidity, firmness, shape, maintaining of shape, and then in movement, the kind of restricted movement in plants. So next we want to discuss the supporting tissues in animals and the benefits. So like plants, supporting tissues help maintain shape, and then the group called vertebrates, they have developed bones in addition to the tissues. These bones help them to stand erect, move, respire, and carry out other life processes. So we'll see how supporting tissues will help in other processes of life. So in animals, the supporting tissues are working hand in hand with skeletons. That's a bony framework of the body that provides support, shape, and even protection. So in animals, generally, there are 3 different forms of skeletal materials found. And these are cuticles, bones, and cartilages. The picture there shows a dragonfly, and that's a typical example of a cuticle or exoskeleton, which we will discuss later. And then man, you can see the skeleton inside, obviously made up of bones and some parts of cartilages. And then you see a jelly-like structure like Medusa there. And there it also has cuticle as well as hydrostatic skeleton. We will discuss all of this shortly. Starting with the cuticle, it's an exoskeleton that's found on the external part of the body. It's composed of chitin, a non-living substance, and animals or organisms that have cuticle and exoskeletons can only grow by molting. And molting means they have to shed off the old exoskeleton first Then the body will be naked and soft. They increase in size and then develop a new exoskeleton or cuticle. So the picture there shows an insect coming out of its former skin. You can see how transparent and fragile it looks. But that's to enable it grow or increase in size briefly. So that process of molting is also known as ecdysis. The second type of skeletal material found in animals is bones. They are made of living cells known as osteocytes, and also protein fibers called collagen, and minerals like calcium phosphate and calcium carbonate. All these make up bone. So the presence of the non-living component or minerals fortify bones and make them stronger and more rigid than cartilage. That's the structure of a bone there, and it consists of a hard outer layer. You can see the outer layer there, or shaft, and a spongy or hollow cavity filled with bone marrow on the inside. Structure of a typical bone. Here are examples of organisms with bony skeleton. You should know this already. We have a giraffe there. You can see how it looks, how its skeletons look inside. We have a frog, an elephant, a crocodile, and a dog. They are all made up of bony skeletons. Now, cartilage is another skeletal material that is commonly found in cartilaginous fishes like sharks, rays, but it is also found in mammals. Parts of the skeletal system in mammals is made up of cartilage. So mammals have both bones and cartilages. Now, cartilage consists of living cells also, but they are known as chondroblasts. And in addition, it contains some carbohydrates and protein fibers. Here are some organisms with cartilaginous skeletons: the shark, ray, the skate, and the ratfish. And like I said before, mammals also have cartilage mixed with bones in their skeletal system. There are three types of cartilage that are found in the animal kingdom, and these are hyaline cartilage, fibrocartilage, and elastic cartilage. Hyaline cartilage is found in the trachea and bronchi, trachea around the throat, bronchi leading to the lungs, and on the surface of movable joints, and also on the protruding part of the nose. That's hyaline cartilage. Fibrocartilage, though tougher than hyaline cartilage, is also found in small bones and in the vertebral column of backbone. Elastic cartilage is found on the ear. That's why your ear can be very soft and flexible. It's made up of elastic cartilage. Here is a diagram or a picture showing the different internal structure of the three types of cartilages found. There are some differences between bones and cartilages which are here for you to study. So you can see bones are firm and brittle. Cartilage is soft and elastic. Bones have blood supply, but cartilage don't, and so on. So I will advise you study and know the difference. Now, coming back to skeletons, we did mention that there are different kinds of skeletons found in the animal kingdom. So here we've listed the three major types. So skeletons may be endoskeleton, that's found on the inside of the body, like in man, or exoskeleton, like we see there on a snail, in insects, and so on. And then we also have what is called hydrostatic skeleton. Pressure from fluid gives them firmness and support. So it acts, pulls on the muscles to like skeleton. So for hydrostatic skeleton, they are found among soft-bodied animals, and it's made up of fluid that fill the spaces of the animals, and this fluid is incompressible, so they press against the muscular wall, causing muscles to contract and exerting pressure just like bones do. And so this kind of support from a fluid gives rise to hydrostatic skeleton, maintaining the animal's shape and enabling it to move from place to place. It can be seen in earthworms and other soft-bodied organisms like sea anemones. Exoskeletons is another type. We talked about it when we talked about cuticle, because it's found exo, it's on the outer part of the organism. And like we said, most animals, invertebrates especially, possess cuticle, which is composed of chitin, which is a non-living structure. We talked about growth being taking place only when the exoskeleton is shed, and then that is by molting or ecdysis. So, third type of skeleton is endoskeleton. It is found on the inside of the body of animals, and it may exist either as a bony form or cartilaginous form, like we've said before. And in vertebrates, it is a mixture of both. So examples include the skull, vertebral column, bones of the hands and legs. Now, talking about skeleton or endoskeleton, for mammals, the skeletal system can be divided into two, and this is based on their structure. So we have axial and appendicular skeletons. So from their name, you should be able to know that axial skeleton is made up of the skull, the vertebral column or backbone, the ribs, the sternum, while appendicular skeleton is made up of every other skeleton outside the ones mentioned above. So from this diagram, you can see here axial skeleton made up of the skull, the vertebral column, just in that line vertically. Skull, vertebral column, the rib cage, all belong to axial skeleton. And then appendicular skeletons are the other skeletons that hang on the axial skeleton. Every other skeleton that hangs on the axial skeleton. That's the shoulder girdle, upper limbs, girdle, pelvic girdle, lower limbs. They are all part of the appendicular skeletons. Now, we'll take axial skeleton first. It said it's made up of the skull and the vertebral column along with the rib cage. So the mammalian skull is made up of several flat bones. These bones are joined together by sutures, and they serve a protective function, protecting the brain and other vital organs like the eyes, nose, ears. And also the teeth is born on the jaws of the skull. Now the skull in itself can be divided into three major parts: the cranium or the brain box, as you can see there from the diagram. It holds the brain. Then we have the facial skeletons, the one that supports the nose, the eyes, and muscles of the cheek. And then in the skull we also have the jaws. We have two jaws. Upper jaw is immovable. It's also called maxilla. And then the lower jaw is the mandible. Both jaws contain teeth fixed in the skull. Now the vertebral column which is the second part of the axial skeleton. It's also known as the backbone or spinal column. It's a supporting structure in the skeleton. It protects the spinal cord, and it is made up of 5 different groups of bones called vertebrae. That's where we get the name vertebrates. And then those without this backbone are called invertebrates. So in humans, the vertebral column consists of 33 vertebrae. But in rabbits, it consists of up to 46 vertebrae. And you should know the reason why. Rabbits have tail. In rats, it will consist of even slightly higher number of vertebrae because of their long tail. So that's the structure of the vertebral column there, and the color coding there shows the different groups of the vertebrae. So we have the cervical vertebrae, which is around the neck region. This is for a man or human beings. So the number there is 7. And then next to that, we have the thoracic vertebrae around the chest region, holding the rib cage. And then we have the upper back, that's the lumbar vertebrae, which is 5 in number. And then we have the sacral vertebrae. So on the right, we have typical structures of each of these types of vertebrae as seen in human beings. So like we said, in mammals, the 5 different vertebrae are cervical vertebrae, which is found in the neck region. There are 7 in man, 7 also in rabbit, 7 also in rats. The thoracic vertebrae is found in the chest region. Man has 12, rabbit also has 12, and we find 13 in rats. The lumbar vertebrae is found in the upper abdomen. So in man, there are 5 in number, 7 in rats, and then 7 in rabbit, and then 6 in rats. The sacral vertebrae is found in the lower abdomen. There are 5 in man, between 3 and 4 in rabbit, and 4 in rat. Now the caudal vertebrae is found in the tail region. And since man does not have a tail, there are only 4 of them, though they are fused. In man, in rabbits you have 16. And in rats, because of their long tails, you have between 27 and 30 of them. Now, there are common features of the vertebrae, though they are in different locations and serve different functions, but they have similar features. So a typical vertebrae is supposed to have these following features. It's supposed to have a neural canal. This is for the passage of the spinal cord. And then it's supposed to have a neural spine. If you touch your back there, you see some projections, bony projections. Those are the neural spine. They project upwards dorsally and is used for the attachment of muscles and ligaments. Then a typical vertebrae is also supposed to have a centrum, that's a solid bone piece, or in some textbooks they call that the body. In addition, it's supposed to have facets, which are small flat areas that make contact with other bones. And then it's supposed to have articular surfaces for the articulation of successive vertebrae. And these are generally known as zygapophyses. So we have 2 of those, the pre-zygapophyses and the post-zygapophyses. The structure here shows a typical vertebrae with all the features that we have described. Remember, this is the typical structure, but depending on the location where they are found, either in the cervical region or thoracic or lumbar vertebrae or caudal, these features may be highly specialized, either modified, reduced, or increased. But typically, a vertebrae should be should look like this. So this is where we will stop for today's class, and I'll see you next time.